Ready? I'll begin. Okay, so I'm Jacyln Bubnell from Wells College, and this summer I did my research on using olfactory placode cell lines to study gene expression of odor receptor promoters. So we're interested in gene expression of odor receptors because they have an unusual way of expressing genes. They use monoallelic expression, which means that each allele is actually treated as a single gene, and only one is chosen by each cell. So it also goes through a really interesting mechanism where it has two chances to choose a gene. So transcription, translation occurs, and the proteins that's produced will either tell the cell to go into a type of maintenance phase where it'll continually produce that protein and it will continue it. So that's what this first picture looks like. This is first choice. And then if the protein says that it doesn't like what the gene told it to do, it'll go through a second choice and pick a different gene. And it has a library of about 2,000 genes to choose from. So that's why you don't see all of the same things going in the same area in that picture. So that's second choice. So in order to study gene expression, we refine odor receptor promoters to study it. And we have an odor receptor promoter region, which drives a marker gene to see if the promoter worked or not. And then we can take out a part of the promoter region and see if we get first or second choice. And if we get first choice, we can do the process again, knock out another region of the promoter, and then go through this process again until we get second choice, and then we know which region of the promoter is necessary for gene expression. So in order to study this, we've been using transgenic mice, but it's time, costly, and expensive. So an alternative to transgenic mice is to use cell lines to study gene expression, and the problem with using cell lines for this experiment is that olfactory neurons are highly differentiated and they're non-mitotic, which means they won't grow in culture. So there's no time to really see what our results are. But lately there have been studies on olfactory placode cells, which are undifferentiated versions of the olfactory neurons. So some information about these cells: they're found in the olfactory neuroepithelium. They exhibit the same monoallelic gene expression that's found in the odor receptors, and they're undifferentiated at 33 degrees, but they can differentiate into mature olfactory neurons at 37 degrees. And the OP cells are kind of caught in this stage in between being mature olfactory neurons, which don't grow in culture, they're non-mitotic, and basal cells, which grow but they won't show gene expression. So theoretically, these OP cells should be in like a good part of their development to show expression. So our hypothesis is that OP cell lines are able to express odor receptor promoters. And this is because they're mitotic, so they'll grow in culture. They display singular gene expression just like odor receptors, and they have characteristics of mature olfactory neurons as far as morphology. So our experiment, we used two types of cell lines, OP6 and OP27, and promoter constructs that we knew what the gene expression looked like in transgenic mice, so we can compare them. So the control constructs we used were D302, which has an odor receptor mini promoter that we know does not drive expression in transgenic mice, and also a reporter gene, so we can see if it turns a color that it worked or not. So we used D516, which has a reporter gene promoter. This isn't an actual odor receptor promoter, but it's a promoter we know will drive high levels of gene expression. And we also have a red fluorescent reporter gene, and this one contains a gene for puromycin resistance. We have D301, which has a reporter gene promoter as well, which we know will show gene expression, and the same reporter gene in D302. And this doesn't have an OR promoter either. And then D202, which is just the reporter gene promoter and a green fluorescence marker. So our test constructs, we have D127, which has an OR mini promoter that was shown in transgenic mice to have positive gene expression. D452, which has a very high probability of showing gene expression. It's an OR promoter, and it also contains a receptor gene. It's not an actual odor receptor gene because if that existed and it was put into the cells, they would automatically differentiate and we wouldn't be able to get any results. but it's a receptor type gene. And D454, which has a high probability promoter and drives a yellow fluorescence reporter gene. So in order to get our results, we use a technique called electroporation. So what that is we combine our cells and our constructs together, and then we shock them. And the shock allows the membrane to open up for a while and the DNA to move into the cell. So we used episomal DNA, which is plasmid DNA, so it's circular, and it won't be incorporated into the genome, but we hope that it would be enough time for the cells to express the gene. So we did this first electroporation to see are our OP cells electroporatable, and can they be stained with X-galactosidase, which is one of the reporter genes, so we can get blue staining. And we ended up seeing that D202 showed green fluorescent protein, D301 showed blue staining with XGAL, but we didn't get any expression for the other two. So this meant that our cells are electroporatable, and we can stain them with XGAL. So these are the results of that electroporation. You have positive green fluorescence in the nucleus, which shows that our cells can be electroporated, and you can see the blue staining in the cytoplasm of that cell. So we know that XGAL staining could exist. So we asked the question, why was there no positive blue staining for the test construct D127? This was a test construct that had an OR promoter that showed gene expression in mice. So since the transient DNA is circular and it can't be permanently incorporated into the genome, we decided that we should make stables, which would involve us linearizing the DNA and then electroporating them into the cells. And this would incorporate it permanently into the cell line genome. So we linearized them using restriction digest, and we used D516, our control construct. It has a marker for puromycin resistance, so then we could select for which cells got the DNA by putting them in media with puromycin. So our second electroporation, we used stable DNA, and every cell was electroporated with D516 and then the test constructs. So what we got was that we saw that D516 was electroporated into the cells, but we didn't see expression for any of our test constructs. So this is what this one looked like. You can see D516 has the red fluorescence in the nucleus, and these cells are stained with mitotracker. It's just, it's not exactly, it doesn't represent gene expression, but just so we can see the morphology of the cells. And this is the same thing, but it just doesn't contain mitotracker, but you can see the red nucleus. But we have no gene expression. So we needed an explanation as to why we weren't getting gene expression. So we thought that maybe these cells needed to differentiate before they could express the promoters. This is because they're no longer growing, so they could have time to make a gene choice, like the mature olfactory neurons. And if you recall, OP cells differentiate at 37 degrees Celsius. So we shifted the temperature to 37 degrees Celsius and used the cells from our previous electroporation, and we left it like that for 3 days, and then we imaged them, and we saw that we did get gene expression in construct D452 over here. So this is what the gene expression looked like. We have our red fluorescence from our control in our nucleus, but then you can see that there's red gene expression in the cytoplasm. And this is in comparison with the same cell line at 33 degrees, where you just see it in the nucleus and nothing in the cytoplasm. So these are some more examples of these same results. You can see in the cytoplasm there's gene expression, and over here there's gene expression. So we asked another question, why was there no gene expression in our other constructs? So one possible explanation is that our construct D452 was our only construct that contained this receptor. So the OP cells were able to recognize that there was an OR promoter, and they saw that there was a receptor-like gene. So it expressed it, it created a protein that then told it to go into a maintenance stage and continually make that protein. But in the other constructs, there was no gene for a receptor. So the cells, they didn't really like to be tricked, and they saw the promoter there, they didn't see that they were making a receptor, so there was no use for them, and they made a second choice. So for future directions, we can perform further experiments to enhance our knowledge of these cell lines and what they're capable of. And we know that they're a viable means to study gene expression of odorant receptor promoters. And this knowledge will allow for further study of the function of OR promoters with a higher efficiency. And I'd like to acknowledge the following for making this possible. And that's it. Any questions?